Hello, welcome back to microbiology. This is going to be chapter 4 covering bacteria and archaea, or really prokaryotes. We can compare bacteria and archaea versus eukaryotes. One of the bigger deals here is that the prokaryotes don't have a nucleus, so bacteria and archaea don't have a nucleus, while eukaryotes do. One big difference between bacteria and archaea is that actually bacteria have cell walls primarily made of peptidoglycan, while archaea will have different compounds making up their cell walls. This makes a little sense whenever you consider that archaea are found in extreme environments, and as such, actually will have the cell walls that tailor or fit the environment that they're going to be in. Other important things here are internal structures. Bacteria and archaea, or prokaryotes, do not have membrane-bound organelles. So, this is an example of a hypothetical cell. As you can see, this image is just trying to illustrate different parts and pieces that we would find in a bacterial cell. So again, this is not any normal real bacteria. We're going to cover most of these parts as we go through. So again, you can take a look at this at your free will, but again, we'll go through each of these parts. So let's point out that bacterial cells are single-celled organisms. Again, these things are capable of carrying out normal life activities like reproduction and metabolism, and they break down nutrients and resources. They can act as a group or as a large mass of bacteria in things like colonies and even biofilms. Bacterial size, as we've already discussed before, they come in a variety of sizes, but overall around the average is 1 micron or micrometer. Some of them tend to be bigger, some of them tend to be smaller, but the average being around 1 micron. There are two major shape types that we'll really focus on in here, and that's cocci and bacillus. A coccus is going to be spherical or round, while a bacillus is going to be kind of like a tube or like a rod. Again, the idea being that these are different shapes. In general, again, there are some cocci, there are some bacilli, but there are a category where there's no consistent pattern, and we call that pleomorphism. If there's no consistent size and shape, then again, we can't call them cocci or bacilli, they must be pleomorphic. So here are some examples of things like a cocci on the left here, the bacillus or rod in the middle, and then a vibrio, which we didn't discuss much, but it's a rod, but it's a bent rod. It has a bend to it. Some other things shape-wise that we should be interested in are the spirilia. So spirilia includes spirilium, which are short, fat, helical cells, and then the spirochete, which are long, skinny helical cells. So the idea here is we have helical cells in spirilium and spirochetes, but spirilium are short, spirochetes are long. Spirilium are fat, spirochetes are skinny. So let's point out that there can be different arrangements for the shapes. So shapes like cocci, so these are just round cells, can come in different types of arrangements like diplo or tetrad or staphylo. The idea though is you should probably know these to help you with your identification in things like the unknown project, as well as on this exam. So diplococcus actually stands for a pair of circular cells that are stuck together. Tetrads are actually 4 cocci stuck together. Staphylococcus are an irregular cluster. Streptococcus is a chain. And then Sarcina is usually a cuboidal packet that increases as a cube. The bacilli are also going to come in arrangements, including things like diplobacilli, or 2 rods end to end. We also see things like streptobacilli, which is a chain of cells. We see palisading cells. So unlike the strepto that are tip to tip, palisading is attached to each other lengthwise. So they look like pickets in a fence. Overall, spirilia and spirochetes don't typically group together, but spirilia on very rare occasion can be found in short chains. So, now that we've talked about shapes and arrangements, let's start talking about some of the structures, including the external structures like flagella, fimbriae, and even pili. Let's actually start off with flagella. A flagella is going to be a proteinaceous extension that allows for movement, but a flagella is actually made of several different parts. Really, three major parts for us. The basal body, which is the molecular motor that allows for rotation. The hook, which is the adapter that allows for power transfer to the large proteinaceous filament located here. So again, the idea here is this molecular motor or basal body turns this very large filament. In general, if you're starting to turn a filament, you want the filament to take a very large sweeping arc. But at some point that tends to create drag. So one cool thing about the hook is it actually starts to allow the straightening of the flagella as speed increases. We also can see flagellar arrangements. So we can have arrangements for the flagella. There are really two major categories, polar and peritrichous. As the name polar suggests, we're talking about one or more in, while peritrichous is going to be found all over. Under polar we have three subcategories, mono, lopho, and amphitrichous. The idea here is monotrichous will have a single flagella. Lophotrichous will have a bunch or tuft, and then amphitrichous has flagella at both ends. So if we look at example A, what do you think it is? Since there's one flagella at one pole, that would clearly be a monotrichous. Notice B. B has several flagella at one end. So at one end means it's polar, but a small bunch or tuft we called, again, lophotrichous. Notice the letter example C here has one filament at each end, but again, since it's an end, it's polar, and since it's both ends, it's amphitrichous. And if you recall, letter D here would be the example of peritrichous, where we have flagella all over. So we should probably talk about what we use the flagella for, which is movement. Interestingly enough, this part of the word taxis actually indicates movement. So whenever you see taxis, we're talking about movement. Since chemo precedes taxis, well, what do you think that means? What are we moving in response to? If you guess chemicals, then you're correct. So chemotaxis is movement in response to chemicals. But you can be moving towards a chemical called positive chemotaxis, or away from a chemical called negative chemotaxis. So really at this point I care that you know taxis is movement, chemo in this case is in reference to what we're responding to, positive is towards a stimuli, and negative is away from. So what would you tell me the positive phototaxis would be? So, runs versus tumbles. So far we've illustrated that bacteria can have flagella, and those flagellas can rotate to create movement. But whenever a bacteria actually moves, it doesn't move like you or I. It actually, again, is rather inefficient in movement and honestly struggles pretty extensively. The best thing I can compare this to is like a small child learning how to walk. They can and will get there, it's just going to take more time and effort than you or I will. So really, bacteria move in what we call runs and tumbles. A run is propulsion forwards. What happens though is whenever bacteria needs to figure out where they're going, they try to stop. But instead of slowing down the rotation, they reverse rotation, resulting in the whole bacteria tumbling or rolling to a stop. So the idea here is, again, a run is moving forwards by rotating the flagella counterclockwise, while a tumble is reversing the rotation to clockwise, resulting in the whole entire cell tumbling to a stop. So here's an example that illustrates this, and this is easy to kind of see now, the comparison to how an infant or a very young child learning how to walk best shows up here, right? So notice, a tumble is a red circle, a run is a green line, right? So if we look at this first example where there's no stimulus, there's no chemicals, there's no light, there's nothing to encourage movement, notice the bacteria still move. But notice overall there's no general kind of directionality to the movement. It's just kind of meandering around. But notice what happens whenever we introduce a stimulus. Notice how movement becomes more linear. there's a path that we're heading either towards or away from. So maybe we put a negative stimulus over here, causing the bacteria to run to the other side. Or maybe we put a positive stimulus over here, again encouraging the bacteria to come to this side. So there is a unique endoflagella that we should probably talk about that's very, again, unique to our spirochete bacteria. What an axial filament is again an internalized flagella. So it's two or more long coiled threads found within spirochetes. But what happens here is this is important in creating a twisting or torquing of the cell itself, creating rotation. That rotation can actually allow for the opportunity for the bacterium. The next thing that we should probably talk about are those additional appendages sitting on the outside, like the pilus and the fimbriae. The fimbriae are going to be small, bristle-like fibers made of protein that extend beyond the surface of the cell. These do not typically move. These are actually really there to be kind of like spikes or projections. But what happens is these spikes or projections can get stuck to other spikes or projections, including other bacteria, like we see in example A here. Or even in the intestinal tract here, where the projections get stuck in our microvilli of the intestinal tract, allowing for attachment. We haven't discussed it yet, but overall the idea is attachment can lead to a greater chance of infection. So by having fimbriae, something is actually more likely to make you sick. Pili are the next topic we should probably tackle. Notice that they're going to try to call it a sex pili here. I'm not a huge fan of that. That's because sex in biologic sense is used for reproductive purposes, while a pili is really not. Pili are actually used for conjugation, or the exchange of genetic material from one cell to another. This doesn't make a cell that's going to be a hybrid offspring. Instead, it just makes two cells more similar to one another. And as a result, again, I don't like the term sex in reference to a pili. The glycocalyx is the next thing we should probably discuss. A glycocalyx is going to be things like a slime layer or capsule. There is a difference between these two. A slime layer typically is going to be loosely attached to the cell and pretty random in the way it's put together. Really, it's there to prevent water loss and, again, loss of nutrition. While capsules, on the other hand, are going to be very tight. They're going to be very dense, thick, and really protective. A capsule can actually even protect against things like phagocytosis. A slime layer is going to, as you can see, be again loose and sloppy. So you can see again there's lots of space in here, and there's not much consistency or pattern to it. The capsule, on the other hand, is again very dense, very well packaged in such a way that whenever a phagocyte tries to grab a hold of the encapsulated bacteria, it actually has a hard time gripping it, much like you trying to hold a bar of soap in a shower. So you can see again the examples here. So this is that capsule stain that we've seen previously. So I can see the bacteria in the middle. I can see the negative staining in the background. What lies in between here is the capsule itself. Notice down here, even at macroscopic level, you can actually see differences in the glycocalyx producing bacteria. As we already said before, capsules again prevent phagocytosis. So it's important to understand that this would make a bacteria more dangerous or more likely to cause infection, thus increasing its pathogenicity. Biofilms are something we haven't got to talk about yet. Biofilms aren't just one bacterial type, but often many bacterial types living and working together. The best example I can think of is like a commune. The idea here is pretty simple. These microbes are going to interact with one another and support one another, allowing for a growth of a very large community. But one thing we haven't mentioned is that very large community is going to dump essentially lots of sugary materials that surround all the bacteria that are present, thus kind of protecting them or barring them from the outside world, preventing chemicals from reaching them, and ultimately making them very hard to treat in medicine. So you can see here with the formation of a biofilm, it typically starts off with some initial colonists developing. They stick in a special place, and then they start to develop things like a glycocalyx. And before long, you have this huge mass of sugary material with all these different bacteria living in that mass working together. Again, they'll work together to help really keep each other alive. The cell envelope is going to be an important thing that we should discuss. The cell envelope is going to be made up of essentially 2 or 3 basic layers. the cell wall, the cytoplasmic membrane, and for some bacteria, an outer membrane. It acts as a basically centralized protective unit, and so the idea here is gram staining really has a major role in showing us the difference in these cell envelopes. So let's discuss gram staining for a bit. Gram staining was again developed in the 1880s by Hans Christian Gram. He really again recognized there were really two major groups here. Bacteria that had a cell envelope made of a single cell membrane and a very large cell wall, or gram negative that ended up with two separate membranes, an inner and an outer membrane, with a very small thin wall in between. So we can see that visualization here. Notice each of them do have a cell membrane. All cells have a cell membrane. But notice the difference in the cell wall. The gram positive has a single very large cell wall, while the gram negative has a single very small cell wall. And it's because the gram negative invests in producing another membrane, an outer membrane, that helps protect it. Okay? And this actually works quite well. By having two membranes, there's two levels of screening that can prevent things from getting through. With just a giant cell wall, things often get by. And as a result, gram negatives are far more hard to kill and far more dangerous than the gram positive counterparts. If we destroy this thick cell wall, the gram positive is likely to die. The cell structure, as we said before, of the cell wall is going to primarily be made of peptidoglycan. Peptidoglycan is by far a common feature in bacteria, and it's made up of a very unique macromolecule basically composed of glycan chains cross-linked with short peptide fragments. So essentially peptidoglycan, peptides and sugars. And it's important again that it provides this network that gives a strong but yet flexible frame. So you can see this pattern here. We have really the two parts, N-acetylglucosamine and N-acetylmuromeric acid. The idea though is these things need to be linked together through things like cross-link bridges. Those cross-link bridges is what gives the structural support. Interestingly enough, this is what can be damaged whenever you take antibiotics like penicillin. Penicillin actually directly affects the bridging, preventing again that structural support needed. And thus, whenever a cell comes under osmotic pressure changes, it can actually then lyse or explode. And that's really how antibiotics like penicillin work. The idea here is again by damaging that cell wall that the bacteria are so reliant on, it can actually result in the death of the cell itself. So as we mentioned before, the gram-positive cell wall is again very thick, lots of peptidoglycan and lipoteichoic acids or teichoic acids. The idea here is again we're investing in a thick cell wall. That's our strategy for defense. The gram-negative, on the other hand, has a much smaller cell wall, only 1 to 3 nanometers, and it's because instead we're going to invest in a secondary outer membrane. So instead of having a big barrier, we're going to have two levels of security to help control what moves across. There are non-typical cell walls that exist out there. So instead of having, again, the consistent features of a gram-positive or gram-negative, some bacteria actually invest in developing a mycolic acid layer. This again helps out in basically loss of water, which allows again for increased survivability in places where desiccation are likely. It's important to understand though, since we have this mycolic acid basically barrier, that gram staining typically doesn't work for non-typical cell walls. And as such, we had to develop a specialty staining technique called acid-fast staining. So acid-fast staining is really all about staining mycolic acid bacteria. Mycoplasmas are unique and important to us in that they don't have cell walls. They're one of the only bacteria that typically do not have a cell wall. Instead, what they're going to do is stabilize their membrane using sterols to limit the likelihood of lysis. But since they don't have a cell wall that provides rigidity and structure, they don't have a consistent size or shape, and thus they're considered to be pleomorphic. Gram-negative bacteria do have important features that we should probably point out in their outer membrane, and that's typically going to be something like LPS or lipopolysaccharides. Lipopolysaccharides are going to be used as recognition markers, really primarily from our immune system. They're also going to help stimulate fever and shock reactions because they are in themselves endotoxins. So whenever our body sees LPS or lipopolysaccharides, we view it as a threat and start to have essentially panic from the immune system that could result in shock and death. That's literally how toxic shock syndrome works. Porine proteins are going to be important as well. That outer membrane, we need a pathway for certain compounds to get through, so pores become that pathway. The cytoplasmic membrane or cell membrane is again a common feature across all cells, not just prokaryotes. I want to point out that overall it tends to be around 5 to 10 nanometers. That's pretty flexible, made of primarily lipids and proteins. Believe it or not, there are far more proteins there than there are even phospholipids. So again, it's roughly 60-40 in terms of protein to phospholipids. Mycoplasmas, as we said before, instead of having a cell wall, would invest in putting more sterols into the cell membrane to offer them protection. Cell membranes tend to be very important to all cells because this can be a site for things like energy reactions, nutrient processing, synthesis, and even transport. So naturally, since this is so important, all cells, not just prokaryotes like bacteria and archaea, actually have them. One of the bigger things we mentioned there was transport. It's important to understand that a cell membrane is going to be selectively permeable, meaning that we're going to control what's capable of moving across. Some of the only things that are really freely allowed to move across is water and small uncharged molecules. This is important in understanding how transport actually works. The outer membrane and gram-negative bacteria make them basically impervious to antimicrobial compounds because we can't get them to the structures we do usually affect. The reason gram-positives are much easier to kill with antibiotics is many antibiotics actually target and destroy the peptidoglycan layer. But since there's an outer membrane beyond the peptidoglycan layer in a gram-negative, we can't actually get the drugs where they need to do their work, and thus we can't typically kill gram-negatives with those style of drugs. The cell envelope, as we said before, is going to be made of a variety of things, but those things can actually interact with human tissues and cause problems. Proteins in that outer cell wall of gram-positive bacteria can be toxic, so we can actually view the gram-positive cell wall as being a threat and causing immune response. Lipids of mycobacterium are also recognized by the body as being a threat and also again cause immune response. Another important thing we should point out is macromolecules in the cell wall are seen again as being foreign and stimulate antibody production and immune response. So even the presence of peptidoglycan is being recognized as being a threat, and as such, those outer features of the envelope promote immune response. The bacterial internal structure is very important to discuss, but there's so much potential in what we could discuss. So let's start off with kind of the liquidy component that makes up the cell, and that's the cytoplasm. The cytoplasm is typically considered to be gelatinous, primarily made of water, with some dissolved resources like sugars and amino acids and salts and such. We also see things like chromatin, ribosomes, granules, even cytoskeleton that are important for the cell's function. Another important internal structure we should discuss here is the bacterial chromosome. One major difference between bacteria and eukaryotes is bacteria tend to have a single chromosome. And in this situation, again, they're going to be circular instead of linear. If you recall, humans should have 46 linear chromosomes, while bacteria should have one single chromosome that makes a circle. These things are actually going to gather in an area that we loosely refer to as the nucleoid region. Again, this is not a membrane-bound structure. This is not a nucleus. This is just the region in which the DNA is going to be highly compacted. Plasmids are going to be another important feature found commonly inside of cells. These plasmids are actually non-essential chunks of DNA that again can actually be passed through things like conjugation. So it's important to understand, like the pili example in conjugation, that we wouldn't pass our whole chromosome to another cell. We would instead pass these little tiny rings of genetic material we call plasmids, so we can copy them and pass them to our neighbors. Another important thing about plasmids to understand is these things are often not necessary for core. Ribosomes are something that's going to be consistently found. As we said before, ribosomes are used for protein synthesis. Naturally, if you're going to be a cell and you're going to live, you need to make proteins. So having ribosomes is crucial. Even bacteria have ribosomes. But one big difference between bacterial ribosomes and eukaryotic ribosomes is the difference in size. Bacterial ribosomes are considered to be 70 S, S being Svedberg units. This is a measurement basically of how far something moves whenever it's spun really fast in a circle. And so the idea here is structurally the bacterial ribosome is a little smaller than the eukaryotic ribosome, which is 80S. Inclusion bodies are unique storage sites for some bacteria. Again, whenever we have an abundance of resources, we may want to hoard things. Some bacteria hoard things like metals, other plastics, some even hoard vinyl, much like a hipster. The cytoskeleton is going to be important for all cells that exist. The cytoskeleton is going to typically be made of a whole lot of proteins that provide structural support. There are really three levels of proteins associated with the cytoskeleton of a cell. Actin, which are small, intermediate filaments, which are kind of middle of the road, and then microtubules, which are much larger. The endospore is an important thing for survival. Not all bacteria can form an endospore. In fact, endospores are really produced whenever situations get bad. So really, bacterial endospore formation happens whenever things become basically not good. So this is a survival mechanism. A good example of something you may be able to relate to is the panic room idea. You may live freely in your house when things are good, but if things suddenly become bad, it may be necessary to retreat and hide out in something like a panic room or a storm shelter. The idea there is that inert situation allows you protection, but you also can't carry out normal activities like vacuuming your carpet or cooking in the kitchen if you're stuck inside basically your panic room. And so the idea here is endospores are inert. They can't do anything. They're just resting, waiting until things get better. Once the environment gets better, we can come out of the panic room and become a normal active cell again through a process called sporulation. So you can see here endospores actually resist a variety of things, including heating, drying, freezing, radiation, and even chemicals. So what we're getting at is an endospore is a very effective survival mechanism that really just two genera of bacteria can produce, Clostridium and Bacillus. The endospore, as we mentioned before, comes about whenever there's a problem with the outside environment. So if there's depletion of nutrients, especially important ones like carbon and nitrogen, we may have to form a spore until we can actually find more of those resources. So naturally we see a cell go through sporulation where it develops that spore. That can take several hours to actually do, where instead of working to divide the cell, we're working to reinforce the genetic material and protect it. Whenever we talk about the sporulation process, it acts like basically the beginning steps of normal cell division. So you see we'll copy the genetic material and we'll start making what looks like it could be a new cell. But instead of the cell actually dividing, we're going to start putting layers and layers upon the genetic material and basically form the endospore inside of the bacterial vegetative cell. Once the environment gets bad enough, the rest of the cell can actually go away, leaving just the endospore. That endospore is again inert, not doing anything. Once the environment gets favorable again, we can see germination, where a new vegetative cell comes out of the endospore. Germination of the endospore again is that coming out back to a vegetative state. Typically again, this happens whenever the conditions become favorable again, like the addition of water or again the correction in the environment. And usually again, water is required to help kind of break this down, stimulating usually a lot of enzymes that break down that outer cortex. So why does this really matter? Well, unfortunately for us, there are significant numbers of pathogens that actually have endospores. And again, they come from the two genera, Bacillus and Clostridium. So again, things like Bacillus anthracis, which causes anthrax. Clostridium tetani, which causes tetanus. Clostridium difficile, which causes pseudomembranous colitis, which many people just call C. diff. We see Clostridium perfringens that cause gas gangrene, and then Clostridium botulinum that causes botulism. The reality is endospore-forming bacteria are incredibly dangerous and cause significant disease. Basically, endospores, again, are constant concerns, and thus sterility and cleanliness are very important, especially in the medical setting. Again, things like boiling water, using soaps, disinfectants, that typically doesn't work for endospores. I do want to point out that these often can contaminate cultures and give us inaccurate readings. And naturally, again, in the hospital with open wounds, we have the potential of these pathogens getting in. So the reality is endospores end up being a huge headache in medicine and something that we work very, very hard to eliminate. Archaea are something that we should probably briefly talk about, but much like the example of algae, archaea rarely, if ever, cause infection. And it's really because archaea typically live in extreme environments, and no matter how much you tell yourself, you are not extreme. And as a result, archaea usually don't grow in you. So whenever we talk about some features, believe it or not, archaea actually are more closely related to eukarya than even bacteria. They share similar features in terms of their ribosomal RNA or their ribosome structure. They again do have more similarities in terms of protein synthesis with, again, domain eukarya. Some other things to note, though, is typically they're going to have unique methods in making cell walls and cell membranes, and again, things like the way they compact their DNA and such. So overall, they are pretty different. Even though we often group them with bacteria under the umbrella of prokarya, they're definitely their own unique group, and that's why at a domain level, we see the three domains: bacteria, archaea, and eukarya. So you can see now a nice visual comparison of these. Again, you can take some time and look these things over, but the general idea here is traditionally, although both bacteria and archaea are prokaryotes, they're pretty different from one another. As we said before, archaea tend to be far more similar to eukaryotes and share, again, many more features with them. So we do want to point something out here, in that archaea are most likely some of the earlier examples of what most likely the original cell looked like. Again, archaea are typically the most primitive in terms of their structure and function, and as a result, again, we think archaea may have come up more closely with, again, the original ancestor. They do live in those extreme environments, like the earth was more consistently, again, much hotter, much saltier, much more acidic, things like that, and have greater capabilities of surviving in that. Thus, again, we think archaea came about more likely before the other domains, but at this point we still can't say because we weren't there to see it. So one interesting thing to note here is that they can actually produce methane. So they're well known for converting CO2 and hydrogen gas into methane gas, CH4, which is interesting because, believe it or not, methane has an incredibly foul odor. And so this is basically done through anaerobic respiration processes. So this happens deep in usually aquatic environments. And that's why, again, places like the swamp, where, again, moist mud, again, has a very stinky smell. In fact, methanogens even have set up shop in some digestive systems of animals, resulting in methane production. Halophiles are interesting. So if you don't know, halo means salt and phile means to love. So really, halophiles love salt. Notice before halophile, though, we have extreme. So we're not talking about these things like to grow in a little bit of salt. They like to grow in a lot of salt. So they can actually multiply in 36% NaCl, or sodium chloride, which is incredibly salty, resulting in, again, a unique environment that most other things would not be able to survive in. So we find these in very unique places, things like inland seas, salt lakes, salt mines, even in, again, salted fish. So the idea here is this is typically going to be something that is pretty unique and, again, lives in an extreme environment, as archaea are well known for doing. So you can see that red pigmentation and association with the photosynthesis that can be done. Psychrophiles and hypothermophiles are two things we should discuss. this is really basically extremes in temperature. If you like it really cold, you're crazy, right? Or psycho. So an easy way to remember psychrophile is they love extremely cold temperatures, very low temperatures. In fact, typically close to freezing. Hyperthermophiles, as you can imagine, thermo is for heat, and hyper means, well, hyper, much higher. And so the idea here is hyperthermophiles grow in high heat. They typically survive in basically around boiling conditions. And so the idea here is these archaea can live in very extreme environments, even in terms of extreme temperatures. So typically, whenever we work with archaea, they're incredibly hard to culture. It's almost impossible. So usually we have to use genetic means of recognition. So using rRNA sequencing and genetic sequencing, again, allows us to view identity. But again, we know a little more or a little less about archaea because they are so difficult to work in a laboratory setting. So whenever we talk about classifications, I do want to point out classification is important. So using things like genetics is also very important here, and that's really what's going on. Again, we're blowing up and reworking the classification system, focusing on rRNA or ribosomal RNA. And this is helpful in that we can now start recognizing identity and also using it to differentiate between things and even establishing relatedness. So the idea here is that using rRNA, we get all the pluses that we can't gain from metabolic tests. So naturally, again, characteristics that help us in the understanding of things like evolutionary history, and things like shape, arrangement, growth characteristics, even habitat were things we used to tip us off. But nowadays, again, things like biochemistry in terms of composition, genetics, as I just stated before, or even, again, traits at a molecular level are very important. So evolutionary relatedness is typically the most accurately determined through that ribosomal RNA sequence like I talked about before. Again, rRNA tends to be very stable, and as a result we can see where their changes have occurred and where those changes most likely happened. So again, genetics has really totally reworked our classification system, allowing us to understand more in terms of evolutionary history even. So you can see here again this image of evolutionary relatedness. Again, we think again the original cells, we don't know what they were, but they did give rise ultimately to things like archaea, bacteria, and eukarya, our three domains. And from there again, domain eukarya turned into things like plants, animals, fungi, and then protista.